Okay, continue vision with slide 60. And now we're going to actually talk about the actual sensory transduction event. How do we convert light into action potentials? And the first thing that happens is that when a photon of light hits, say, a rod, it's bleached. The bleaching is converting what's called cis-retinol to trans-retinol. This is like similar to cis trans fat and cis fat and trans fat. It's just a different difference in shape. So retinol has a particular shape in the dark called cis. It's converted to trans-retinol when a photon hits it. This separates the retinol from the opsin because it's a different shape. The opsin now activates an enzyme that destroys cyclic GMP. And the loss of cyclic GMP closes ion channels and turns off what's called the dark current. This is something completely bizarre, again, in the sense system. Your photoreceptors are depolarized. That is, they're turned on in the dark. In order to see, you have to turn them off. And that's what photons do. It closes ion channels, hyperpolarizes the rod cell, and this stops release of glutamate onto the bipolar cell. Glutamate is an inhibitory neurotransmitter. In the dark, the photoreceptors, when they're on, are inhibiting the bipolars, so there's no signal goes on. When you hyperpolarize the rod, when the light comes on, you release the inhibition of the bipolar cells, and this will activate the ganglion cell. And the ganglion cell will fire an action potential and send an AP into your brain via the optic nerve. This slide, slide 61, shows the actual shape of the cis-retinal versus the trans-retinal. So one rhodopsin, cis-retinal absorbs a light ray. Cis-retinal converted to trans-retinal. The trans-retinal separates from opsin. Opsin becomes activated. It's the bleaching reaction. So the trans-retinal leaves. The opsin then goes, inhibits an enzyme that activates an enzyme that breaks down cyclic GMP. Cyclic GMP keeps sodium ion channels open in the dark. And then trans-retinal is now sent into the pigmented epithelium. So this is one of the real purposes of the pigmented epithelium. It converts trans-retinal back into cis-retinal and reassociates the cis-retinal with the opsin to form rhodopsin, and it's put back into the membrane, and it goes through the whole cycle all over again. And this process here of converting trans-retinal to cis-retinal takes about 5 minutes. Okay, slide 62 shows you the same thing again. In the dark to the left, NA channels kept open by high levels of cyclic GMP. Sodium rushes into the cell, depolarizes the cell, and this is called a dark current. A little bit of potassium leaves, but it's still depolarized. Then so down at the base of the photoreceptor, calcium channels are open because the cell is depolarized. Releases inhibitory neurotransmitter onto the bipolar cell, turns off the bipolars, and these calcium channels are closed, and nothing's going on in the ganglion cell. Turn on the light. Rhodopsin is bleached, leading to a drop in cyclic GMP. The opsin turns on an enzyme that breaks it down. It's dropping GMP levels, close sodium ion channels. The ion channels are closed. Dark current stops, but the potassium is still going out, so the cell is hyperpolarized. This closes the ion channels, prevents the inhibitory neurotransmitter from being secreted onto the bipolar cell. Bipolar cell is turned on. Its calcium channels are open, releases excitatory neurotransmitter on the ganglion cell. Ganglion cell is stimulated to threshold, sends a nerve signal into the optic nerve. So here is the truly bizarre thing. In order to see, you have to turn off your photoreceptors. It's bizarre, but that's the way it is. Slide 63 talks about the recycling of photopigment. Transretinal converted back to cisretinal and reunited with opsin to regenerate rhodopsin. Talking too fast. Takes 5 minutes for 50% of rhodopsin to be regenerated, but regeneration of photopsin takes only like 90 seconds. So it's really quick. And this is going to, and we'll see the importance of this in what's called light and deep dark adaptation in the next video. And then optic nerve signals. Remember, ganglion and amacrine cells are the only ones that generate action potentials. Photoreceptors and bipolar cells do not. They only generate gradient potentials.